Genomics, and the topic of our talk is modeling biology with Grakn. I myself, I'm a biologist. My colleague Muhammad, who's going to talk to you later, is an engineer. We will approach this question from both angles, and I will give you a bit of introduction of the biological question and also a bit of background on the company, and then we will dive into our implementation, some of those detailed questions. So as I said, Muhammad is here, who's our VP of Innovation. I myself, I'm Sven, and I head the Bioinnovation Department. So actually, as you just saw in the intro, we've been working with Eagle for quite some time, and if you even go onto their website right now, you will see a quote from our CTO, Rad Udriri, exposing the virtues of working with Grakn, and we've been very excited, and it's been a very interesting and exciting journey together. So who are we? So just a bit of background. This is Alexander Fleming, who is not a member of Eagle, but he, of course, together with Louis Pasteur and Robert Koch, defined our understanding of health and well-being that we currently have, which is also now currently changing. So he invented or discovered penicillin fortuitously over a weekend in a Petri dish. And of course, biology is not like that anymore. Biology, even during my PhD, has changed dramatically from being a wet lab science, which it still is, but has been transformed now by these machines into being a data science. And so nowadays, in many cases, the real value lies in understanding and evaluating the data. So these machines can produce millions of sequence reads every day and every run, and we are challenged by interpreting them. So just a brief overview of our product. I won't go into too much detail. It is a modular product which we have different components, from curation to a catalog, the basis of which is Grakn, through to data valuation and exploration and analysis, going from big data on one side, taking the journey to insight. So that is what we're here to achieve, and I will talk a bit more about the nature of biological information in our context and our focus. So the focus of the part that I'm most interested in, and that we will talk a bit about more today, is actually the microbiome. Now the microbiome, for those of you who don't know, or even just as a reminder, is the collection of bacteria, archaea, fungi, and viruses that will inhabit a certain area of an ecosphere, such as our gut, or our oral cavity, or the soil, or whatever. So that means that there is a human microbiome, or even several human microbiomes, depending on the location of the body. There's also a plant microbiome, or several plant microbiomes associated with different areas of the plant or in the soil. And there's an animal microbiome, or several animal microbiomes in the guts of all these animals, or in their skin, or in different parts. And each of these is highly interesting to us as a company. It's interesting scientifically, but it's also highly interesting commercially. So the interest in the data from these sources has really increased dramatically, and across the sphere and our clients and within the scientific area, people are really just beginning to understand the wealth and richness of these data sets and of the functionality and of the capability that lies therein. For example, there have been enzymes discovered that can degrade plastics from a bacterium, and people are interested in commercializing that. Just one example of hundreds. Okay, so yes, and there's even an air microbiome. So in the case of the human microbiome, what are the current applications and the focus? It has to do with health and disease. So many diseases are thought to be related to what happens in our gut, or in different areas of our skin, or in our other parts. There's incredible interesting links even to Parkinson's, the onset of Parkinson's disease, mental diseases with gut health. There's the area of mental health. There's sort of functional foods that improve mental health. There's links between the gut and the brain. Even in fitness, recovery is affected by your diet. So there's a big growing interest in how your microbiome influences how well you feel and how fit you are in sports or in other areas. And in the pharma sector as well, the way you metabolize a drug determines on the gut microbiome and the functional capabilities that exist in your gut. And so there's an area question of personalized or stratified medicine that has to do with your enterotype, the types of bacteria that you might have in your gut. And of course, food is a large question, so different people react differently to different foodstuffs. It has to do with allergies. It has to do with how well we react to certain carbohydrates, for example, in diseases. If you're diabetic, different people react differently to the same source of carbohydrate, which is mediated by the gut microbiome. And this is just one of the areas, and there are several others which are highly important and highly interesting. So this means the industry verticals that this is interesting is personal care, cosmetics, nutrition, and agri-tech. So this just as a bit of a background as to the wide applicability of the interest in the microbiome. And recently they've been advertising it with this little logo where they say it is microbiome gentle. And we only recently, after the fact, found out that this claim was actually done using our software. So this is about the skin, about healthy skin. So we were very chuffed by that, and we're active in a number of these industry verticals across different areas, as you can see, and we know that this is a growing and extensive area of commercial interest that we work with, either now or in the near future. Okay, so one area is to arrive at marketable benefits. How would you do that? Across these three large areas, because there's a challenge of the data, there's a challenge of the science, and there's even the challenge of the business model. And now getting to the data part, anyone who's a biologist will know there's not just one type of biological data, but several. For example, there's a transcriptome, which is the active part of the cell. There's phenotypic data, what does a cell look like, or the human look like in the end. There's, of course, genomic data, all the DNA that is in the sample. There's metabolomic data, which is all the small molecules, all the metabolites that we can identify now. And there's a proteome, which are all the proteins. And we also know that this data is, so the challenge here is so broadly called multiomics integration. But what we also know from science and from biology is that these data do not exist in isolation. They're highly interconnected and they influence each other, which means that there's another area of big challenges, which could be to determine causality. Even if one thing changes, it's very hard to determine is that the cause or the effect of a change somewhere else, which means we might want to model these interactions. And finally, this all has a high applicability to business because this data can lead to innovation, product innovation, and is important even for messaging, for marketing, and other purposes. So now to the microbiome as a challenge, as it appears for a data scientist, and I'll just go in a couple of them and see how we then try to attack them. The first one I mentioned is multiomics integration. The second one is, that means different data types of different data, different experiments have to be integrated and made sense of. The data in the microbiome sphere is really hyperdimensional. I don't think it's, I'll come to it in a minute. It is the number of variables in these bacterial environments is immense. And if you if you take the connections between them, it becomes even more than astronomical. And another part of this data is that it is compositional. That means we're often not measuring absolute data, but relative data. So the challenges in multi-omics integration are fairly well established and well known in the mathematics field or even in the science, and is that there's a challenge of normalization. Different data sets were derived by different methods. We have to deal with that variety. Then there is something that every scientist knows and every scientist struggles with, is that the metadata is often incomplete. And the other thing that, why I like this topic really a lot, is that it makes the link between data and now annotations, which are semantics, which now we're getting towards a semantic graph model. So we rely heavily on the metadata. This is linked to there's different standards and different levels of annotation that these data are governed by or sampled at or reposited at with. So we struggle with incompleteness there, or different ontologies. And of course, there are confounders that might go unnoticed if we aren't careful with our statistics. I mentioned hyperdimensional, and I don't think this is an underestimation, because the conservative estimates say that in our gut there's at least 1,000 different bacterial species. Now, several hundreds of them have been completely sequenced and are known, but we also know that we don't know a lot of them. So in a conservative estimate, we have 1,000 different bacterial species in our gut, each one of us probably a different subset. Now, if we wanted to just calculate the number, just the mathematical number of different ways they can interact with each other, because bacteria they don't work in isolation. One bacteria might produce something that the other one consumes, or one bacteria might produce something that inhibits the growth of the other. So these interactions are very important. And if we were just mathematically trying to establish how many different types are there, the number is 1,000 factorial. So which is 10 to the power of 2,567. So it is truly astronomical, beyond astronomical. So this means these interactions don't just go between the bacteria, but they involve metabolites, signaling molecules, which then interact with the host, us, and induce a reaction in humans. So understanding this complexity is a huge challenge. And the initial models so far, the mathematical models that exist, aren't really well equipped to deal with these numbers. That results in a vast parameter space, which exceeds everything we can just imagine. Okay, so the other thing is that it's actually also a different paradigm to combat disease, because as I told you in the beginning, the existing paradigm that most pharmaceutical companies or other companies work under is that the bacterium is the enemy, right? You kill it with an antibiotic or with soap or with a detergent or with something. Now that does not work in the microbiome. You cannot eradicate the microbiome. So this works for polio or measles or smallpox, where you can eradicate them completely, but it doesn't work in the microbiome because we would be dead. So the only solution is to try to modulate it. So a little image on the left is supposed to show you. This is a cartoon of as we call it, an abundance graph, where these different colors would indicate the abundance of one bacterial species. And we'll have different bacterial species, and some will have at very low level and some at very high level. And these two states potentially could be a healthy and a diseased state. Now the question is, how do you get from state A to state B? And so what you see is maybe you want to increase the number of this little green bacterium, but we don't know why. How would you even increase it? Because this green bacterium might be inhibited by the red bacterium, or it might grow on top of the blue one, or we don't know. So all these interactions are very complicated, but the question is, how can we modulate this to get you from a disease into a healthy state? So this is a completely different paradigm to work under, other than to kill whatever you see. So we need to understand the interactions in these associations. So this is what I said. Now we're starting to get into the data model of this, is because what we know already is that many of these things. So we know that there's a large set of sort of annotated data, and we would try to understand this in a more structured way, and how do key metabolites, for example, or key gene products interact and help the growth or inhibit the growth of either desired or undesired bacteria. And also, so these are the interactions between the bacteria, across or between species. So this is what we call metaclades. And all of this happens within a context, where the context is the host in this case, if we're talking about human microbiome. As I said before, these metabolites are some things that we take up and we react to, and our gut reacts to, and elicits a response in us. So now we see that we're going across domains. We have genomic data from the bacteria, we have metabolomic data that we measure in the gut, for example, and we have genetic or human data that we need to integrate, and potentially even with human genetics across different regions and the like. So, as we see, this data is highly contextual and highly interlinked. And our approach to this was to say, okay, if we have such a highly contextual interlinked data, why not use a graph language and a graph representation to work with it? And to get into more detail of that, I'll now hand over to Muhammad, who's going to show you some of the applications, and also. Thank you.
Hello. So I'm the computer guy. I'm sure I've already made the day a bit longer for everybody already. So let's go on. And what I'm going to try to do is simplify a bit from what we have. My personal experience has been always about information. I was very much interested in information, and I kind of moved from one industry to another. So telecom, chemoinformatics, that kind of thing. And this challenge, this was a relatively new challenge to me, biology, last 3 years. It was a whale of data, a whole lot of data is a whole lot of context. So my question to you, if you think of it as a whale or an elephant, how do we eat an elephant? Anybody wants to answer? One bite at a time. We just can't do it all together, okay? So here we're going to look at a, no. Okay. Yeah. All right, so let's think about a simpler problem, but let's keep in mind the complexity of the data. And what I mean by the complexity of the data, it's not like what we do in telecom or in physics, where the data is not that contextual. What I mean by that is you have a line that gives you most of the information. Here you touch a gene. Let's look at the gene for an example. We're just talking about a gene, which should be one of the simplest things on the biology. So a biologist enters a laboratory. A biologist tries to look at what we have, and what they have is a scientist is trying to look at the gene. Now, this is within the context of some experiment. We're comparing the genes of two people, one healthy, one diseased, okay, or a case for a certain disease, okay. Now I'm comparing these two, I'm getting a difference between two. So these are differentially expressed genes, a list of them, okay. So what does the scientist want to do? He or she want to find what the pathways in that gene, what disease are linked to those, what are there any drugs targets possible, and a number of issues. And this can be a cycle, because what can happen is if we discover the gene, which is a transcription factor, we want to ask the same question about the genes linked to this gene. So this goes in a loop. The most important thing, and the thing that I'm going to spend the next few minutes at, is basically all of this data changes all the time. That's the fun of biology. Okay? So let's say we've agreed that this gene is linked to this particular disease or drug target. What happens next? What happens is that next day somebody discovers that this link was wrong. Let's delete it. Somebody else discovered that another gene was related to this drug. Let's add it. So that already makes the data very dynamic. Okay. Let's try to kick it up a notch. Somebody says, okay, these genes, we need to describe them in a certain way. Okay, so this is the annotations that we need to do. Okay, so now we've standardized the schema for these annotations. But what happened is that next day, somebody discovered a new way of sequencing the genes, and now we have a new way of annotating. In our experience, you have to have the schema dynamic as well. Okay. Of course, you can always say, let's start from the beginning, and let's work with all the data, and let's put the right model. Okay. And once we put the right model and the right schema, we need to work. Here, our situation is much more complicated because we have to shift the schema. We don't have a choice. The third item is basically scientists are obsessed with evidence. And that is related to the same point. Because when I tell somebody from the database or from the knowledge graph that actually we discover that this gene is related to that disease, the question would be, okay, what's your basis? It was a research last month. Okay, that sounds fine. It was a research 2 years ago. Are there any other researches in the same which gives me some different relationships? Is there an indirect relationship that almost disqualifies the current information? Is there a relation that is stronger and we can utilize? So this is still correct, but we can utilize a different relationship that is more useful. So there is this whole kind of data which is versioned, okay, and the scientists need to be able to look inside. And from my personal experience, people in the life sciences tend to be much more about what I would call interpretability. Think in terms of interpretable ML, machine learning, that kind of thing. They want to know why, okay? They can't trust if they don't know the basics of it. So it makes it very difficult to use things like neural networks without putting some sort of interpretable framework or having some causal framework to give you explicitly what we're doing. And they need the evidence. So to summarize, managing the entities, the relationships between these entities, because we need evidence for the relationships as well, and their version, and the evidence for all of that, with updating the data and updating the schema is our aim. And the reason I say our aim, because it's not a solved problem. We've progressed in it. We had done some joint work with Grakn. Thanks for that. Thank you very much. But we're still finding a few challenges. I'm going to start by a simple example, an example that is solved, okay? And we can look at it and we can see some of the features of Grakn, which helps us, so why we used Grakn, okay? If you look here, the source of the data is captured. So we started by an entity, okay? But we said simply for all our entities, we want to know where the data is coming from. So we're looking at the data source, we're looking at the data URL, we're looking at the version of the data. That should be good enough. Okay. So now we've defined gene happens to be one of a number of what we call sequence features. So we've inherited sequence features from that, we've inherited genes from that. Okay. So now we have this database, we have the knowledge base, we've inserted some data in it. Can we change this just because we've learned that actually we don't do entities one by one. We tend to do sets. So you will have a whole lot of genes, a whole lot of chromosomes that belong to the same data source. That sounds good. In SQL, this is an easy problem. Not easy and is trivial because Grakn, the whole point is helping with, sorry, not the whole point. There is the semantics, which is much more, but it helps with avoiding the just what I call the SQL trap. Okay, but we know how to solve it in SQL with our experience. You do a SER normal form, you know what exactly you're expecting. Okay, so if we go for SQL, what we want to do is we want to put the data source, and this is what we're going to do next. We're going to put the data source in its own relation, in its own table. We're going to have a one-to-n relationship to all the entities, and that is that, okay? Actually, our solution here is not that far from what we're saying. This is abstracted or taken part from our actual schema with lots of fields taken off, but that's the basic idea, okay? The conditions that I forgot to mention is that think of this as database migration while keeping the data intact and keeping the external schema intact. We don't want to update the whole database. Okay. So the trick is simple. First, we defined a relationship has data source. We define the data source. We link the data source to the provenance entity. So we have a relationship has data source. And then we did the same process. That gives me what? All the points, all the genes linked to our data source. Then I put a Grakn rule to make explicit. Think of them if you're used to SQL as calculated fields. Okay. But you'll find that Grakn rule tends to be right than SQL calculated fields in general. So this was our first step. This works. It's good. Let's see what we've solved so far. We've solved the general issue, which can be done with SQL. Let's see our next stage, okay, and this is where I'm going to go a bit off. I want you to scale, each of you to scale this process. I want you to imagine the following conditions. We are going to have to migrate to the next database, changing data and schema every couple of weeks, just for fun. We are going to actually try as much as possible to streamline this process. So if possible, the biologist should write that, not an SQL expert, not a logician. In my dream, not a computer scientist. That makes it much more scalable. Why not computer scientists? Because if we're doing it every 2 weeks, I don't want every biologist writing a paper to go through the whole process. So that's the challenge we're facing, okay. Okay, so I think there is one slide missing, sorry for that. So that's the challenge we're facing. What we're doing right now is collecting a number of what we call Grakn patterns. Okay, so this is the simplest of them, but there are a number of examples we're working with. We've had some help from Grakn on that. What we're hoping to do is to kind of automate the conversion. So think of it as the same as converting from one database, as converting, if you will, from natural language processing, something like that, to Grakn, but using it as near to Grakn syntax as possible. Let me talk a bit about the results, because I think we're a bit risky in terms of time. This is what our system looks like. I know it doesn't, it's not clear some of the semantic aspects, but this is what I was referring to when I said we start. This is a study for a biologist, okay. What they are doing is they are starting by indications, okay. So they're starting this side from the disease or from the phenotype side, going to pathways, going to microbiome. And the way we use Grakn here is this is essentially an interface or an augmented interface with Grakn. Okay, so the backend, the knowledge graph is Grakn. Okay, and this is a process where we kind of go from one node to the other. We navigate a graph that is turned out to be useful because they want to follow the process step by step. Okay. The work we're doing right now, the second part, is basically doing machine learning to propose paths. Okay, so the idea would be the system suggests to use a solution or a direction, but you as a scientist follow what you like. So let me recap here the kind of the whole process. Biology is complex and contextual. If you're interested to understand more about that, people from computer science, I would advise generally to look at complexity theory, okay? It captures a lot of the complexity here. Biology is an evolving science and information is dynamic. That includes the schema as well. Evidence and interpretability is critical. This is a condition we have, and that's kind of the same point, dynamic and complex. And in our experience, biologists and bioinformaticians find it easier to express the science in Grakl, and we're talking specifically about the Grakl language compared to RDF database. Okay, so in biology, there is a lot of emphasis on ontologies, and especially semantic ontologies, okay, in order to capture the knowledge and identify it and make use of it. There is a whole lot of biology in ontologies, things like GO, EFO, etc. But all of these are quite difficult to express and use using updates. So you need to involve a lot of scientists. And in our experience, Grakn was a much more useful and a much easier tool to use. And our head of data modeling is actually a biocurator rather than a computer scientist or logician. So that's one of the main benefits that we got out of the system. All right. Thanks a lot, and I'm happy to take any questions.